Hello and welcome to today's episode of the VJNeurology Podcast. We are an open access video journal providing medical education for healthcare professionals. Today's podcast features a discussion with Professor Jeffrey Dunn on the use of CAR T-cell therapy for the treatment of multiple sclerosis. CAR T-cell therapy is a form of adoptive cell transfer that has revolutionized hematologic oncology and is now being investigated as a targeted immunotherapeutic approach for autoimmune diseases such as MS. Professor Dunn will delve into the mechanism of action of CAR T in MS and share findings from a phase 1 trial. So without further ado, let's go to our discussion with Professor Dunn. Professor Dunn, thank you so much for joining VJNeurology on the podcast. To start, could you introduce the mechanism of action and rationale for exploring CAR T-cell therapy in MS?
CAR T therapy stands for chimeric antigen receptor T-cell therapy, and it's a synthesized, engineered hybrid molecule that's derived from the patient's own T cells, whether autologously or allogeneically, and then often by a viral transducer, such as lentivirus. There's a transduction of the T-cell line to express essentially a B-cell receptor called a single-chain variable fragment. What we end up with then is a hybrid molecule that combines the exquisite specificity of recognition of an antibody to the potent cytotoxic cytolytic action of T cells. So it combines the best of both worlds. As a therapeutic, what this offers is precise target recognition, which should minimize off-target effects, and yet at the same time, potency. With respect to multiple sclerosis, one of the great values of CAR T therapy is that based on the human's own T cells, there is what has been called deep tissue penetration. In neurology, what that means is this molecule, this hybrid molecule that expresses a receptor can get into the central nervous system. So there's central nervous system penetration with CAR T molecules and deep tissue penetration otherwise. So we can get, entrance into lymphatics as well. Given the potential of CAR T therapy, we designed what to our knowledge is the first prospective phase 1 study of using CAR T therapy, in this case with a CD19 binding site that recognizes the glycoprotein that's expressed over some of the entire spectrum of B-cell development, but spares the marrow, which is important for regeneration and reconstitution of B cells. And then on the other end, the terminal differentiation of mature B cells, which would be plasma cells elaborating antibodies, those are also spared because plasma cells don't typically express CD19. But the molecule does recognize the series of maturation steps of B cells, essentially in between those two bookends. And that includes, importantly, memory B cells. So at the same time, while it seems as if there may be great potential for using CAR T cells for neuroimmune diseases in general, and maybe multiple sclerosis in particular, the use of CAR T therapy in B-cell leukemia, B-cell lymphoma, and oncologic purposes has been associated and known to be associated with sometimes very significant side effects, which can include cytokine release syndrome and what's been called immune cell effector-associated neurocytotoxicity syndrome, or ICANS. So CRS or ICANS. And those are neurologic manifestations that occur by essentially abrupt lysis of large cell masses, such as in a CNS B-cell lymphoma. CAR T cells can recognize the CD19 binding site on these B cells that collect in large chunks in these cases, and there can be a significant release of intracellular contents all at once into the central nervous system. That can result in mental status change, encephalopathy, confusion, tremulousness, dysgraphia, word hesitation, and even seizures. So there can be very significant risks. And there have been significant complications seen with the use of CAR T in some neuro-oncologic and neuro-immuno-oncologic indications. So we have a balance. On one hand, the mechanism of CAR T suggests potential in multiple sclerosis and great potential, perhaps, in the treatment of MS, given central nervous system penetration. But at the same time, there are risks that remain undefined.
That's great, thank you. Could you delve deeper into the phase 1 trial and share the findings on the safety and feasibility?
What we designed was a phase 1 study where our primary outcome primarily was to look at feasibility and to look at safety first. We're very excited about these results. It's very important, I would share with my colleagues, the numbers that we're looking at are small. We've enrolled 6 subjects in the study that I'm going to be citing today, although with plans to enroll a large number more going forward. To the first question, is this feasible? The answer is yes. In every case, in every subject that we enrolled, we were able to successfully obtain their T cells by apheresis. We were able successfully to transduce that cell line using a lentivirus vector so that the patient's own T cells expressed a single chain variable fragment on the surface of the T cell that recognized the CD19 glycoprotein. We enjoyed significant expansion so that we were able to infuse patients either with a one-time dose of either 33 million or 100 million cells. And the reason that we used those 2 different doses is that in working with our FDA here in the US, they insisted on a low and slow approach, or what's called a 3x3 design. In essence, they said, try to minimize side effects and potential risks to your consenting subjects. Start with a low dose, in this case, 33 million cells. Watch that one subject extremely closely to make sure they're tolerating things well and there aren't serious adverse events. And if we see evidence of safety, you can proceed slowly. So we ended up in our cohort of 6 subjects to date, as of 2025, with 3 subjects treated with 33 million cells as a one-time infusion and 3 treated with 100 million cells as a one-time infusion. Our protocol was such that we had patients admitted and watched vigorously well in the hospital, checked multiple times for any potential side effects, for any serious adverse events of any kind. And our experience has been the infusions were very well tolerated. There was some mild nausea associated with bendamustine, but not more than that. And once the infusions were given, the patients, frankly, were kind of bored in the hospital because there was nothing more to do. They'd received their therapy. And for the first 10 days after treatment, that none of them really experienced any significant side effects to speak of. Then, on day 10, and this is true for every subject treated, what we observed was an abrupt onset of fever, and it came on quickly. And we know that the fever was associated with the expansion of the T cells that we infused. So those T cells, once they're infused, they function like T cells. They go into the body and they go into deep tissue, including the central nervous system, and they look for targets. They're trained to look for CD19, meaning B cells, and if they recognize that target, the immune system is adjusted so that those cells will expand. And that's what happened. The expansion itself of the cells is associated with an abrupt onset of fever. We only saw one case of all our treated subjects that would qualify for ICANS, and that was what's listed as grade 2, which is mild. The ICANS is thought to be associated with pro-inflammatory cytokines that are released as the immune system activates, thinking that it's getting ready for a fight against a foreign invader. And it's been known we have the great benefit of the giants who have walked in the path in front of us, including those in neuroimmuno-oncology. And we used a medication called tocilizumab, which blocks IL-6, and the patient felt better immediately. But otherwise, all cases were treated with hydration and responded very well. They had fever for 2 to 3 days, and then they returned to baseline, including their baseline neurologic function and their baseline neurologic strain.
You mentioned there was some mild nausea associated with bendamustine. Can you discuss the use of bendamustine for lymphodepletion prior to CAR-T?
When you introduce essentially a foreign receptor on the surface of the patient's own cells, and our study uses T cells collected autologously. There are studies in the future that are planned that will look at allogeneic T cells, and those will be T cells collected from other donors that might partially be matched but would be an off-the-shelf product. Our study used autologous, in other words, the patient's own cells that were engineered to express this B cell recognition site. Because we're giving cells that are not 100% the patient's own once they're engineered, we need to clear some space so that the cells, when they're infused, are not recognized by the subject as being foreign and don't induce a significant immune-associated reaction for the purposes of self-defense. We do that with lymphodepletion. We suppress the immune system just a little bit, again, so that we can clear space, and so these cells reinfused into the patient, which are mostly their cells and recognized mostly as self, still can have space to not be attacked so that they can gain a foothold and so that they can begin to expand so that they can be effective. The primary lymphodepletion regimen that's been used in most studies in this new field tends to use cyclophosphamide and fludarabine, or what's been called Cy-Flu, as the lymphodepletion or conditioning regimen. We didn't do that in our study, for a number of reasons. We used bendamustine, which is an alkylating agent, but bendamustine is, we thought, a little better tolerated, not so much associated with hair loss, so we thought it would be good or more comfortable for patients to consent and proceed. But mostly we did this to minimize the risk of potential cytopenia. Bendamustine, we think, is a little bit more mild than the Cy-Flu regimen. And there have been cases of fludarabine-associated white matter changes of the cerebrum, of the brain, in some of the pediatric literature that's not, to our knowledge, been associated with bendamustine. Finally, one more thing. Both cyclophosphamide and fludarabine is closely associated with another molecule called cladribine that have been used and found to have some impact on the natural history of multiple sclerosis in previous trials. And so we wanted to try to minimize that potential confounder with using an agent that was more specific.
Wonderful, thank you. And finally, are there any further findings from the study that you would like to share, particularly how the patients responded in the months following the infusion of the CAR T cells?
So what happened next? So they were discharged. And I have to say at this point, we didn't know what to expect. This is something that's just not been done or observed so closely before. We don't know the phenotype. We don't know the clinical course. The research that we're doing is intended to provide the foundation for that with information that we can then share with our colleagues nationally and internationally so that downstream studies can be properly designed and patient safety can be optimized. So one thing we didn't expect is that the subjects would feel really run down and experience a lot of malaise. And if anything, I'd say their general experience was that they felt like they had taken a step backwards. In MS patients who have received IV therapy before, some have received steroids, and the use of steroids, especially early in the MS course, can give them a very abrupt sort of next day boost in energy or eradication or partial eradication of symptoms of an MS attack. So I think it could be natural for patients who were just given an infusion of a medicine that they knew was a big deal to feel as if they might feel better overnight. I mean, that's their hope, of course. Wouldn't it be true for all of us? But that's not what happened. They actually felt like their energy levels declined. And they, on average, had about 3 months after infusion, not of serious adverse reactions, no cytokine release syndrome, no ICANS. We monitored blood counts extremely closely, looking at white blood cells, platelet levels, myeloid cells. All of those stayed relatively stable. We followed assiduously for infection and didn't see evidence of secondary infection in that period of time. We have since. But yeah, they felt like they had malaise for 3 months. And then abruptly, just as abruptly as their fever had come on, it seemed as if overnight things turned and they woke up And the most remarkable thing happened. As an important reminder, a phase 1 study, and again, this to our knowledge is the first prospective study that takes on this subject. A phase 1 study means a few patients with close attention to safety. That's its purpose. So I want to stop short of making any therapeutic claims, that you can't make reliable therapeutic claims on the basis of small numbers of patients observed or treated in a single center, not reproduced by colleagues that are widespread, either within the country or internationally. So what we've seen on a clinical basis has to be taken with a grain of salt. But that said, it's been remarkable what we've seen, in my opinion. I haven't seen anything like it in all my years of practice. And I've been at this for a while. So one thing that's important to remember is that the patients that we enrolled in the study were not, in this case, young patients who had just had brand new onset of MS, who had active relapsing remitting disease. There are many, more than 25 approved immunotherapies or disease-modifying therapies for patients of relapsing MS of that type. And so it was appropriate using an experimental immunotherapy to target patients for whom there was no other approved or proven therapy. And so our study enrolled patients between the ages of 18 and 70 who were more advanced in their MS and who didn't have relapsing disease anymore. They were having worsening MS. They were slowly losing strength or balance. They were losing the ability to walk distances or perform their independent activities of daily living. And yet they still maintain function. But in all cases, they had been prescribed treatment that just simply wasn't holding them. Every one of them who came to us said, I'm doing everything the doctor's telling me. I'm still losing ground. I'm starting to worry that the things I love and care about so much, like being able to participate and be a presence for my family, being able to attend my child's soccer games or football games to our European Union friends, but being there for family or being able to work full time, they felt that they might be losing that. So this cohort, in the jargon of our subspecialty, are non-relapsing, worsening cases of multiple sclerosis. That's the cohort that we treated. There were no early cases of relapsing disease. All of my colleagues in this field will recognize at once that this is one of the most difficult therapeutic challenges, because our therapies, such as the ones approved today, it can be said generally they can reduce the risk of new attacks, but the evidence that they slow down or reverse worsening or reverse accumulating weakness from the disease is sparse and sometimes not satisfactory. So to see any effect in this cohort of patients with non-relapsing, worsening MS that hasn't responded to the best therapy we have otherwise is honestly not something that we expected. We hoped at best we might see some stabilization, some leveling off. And the subjects themselves all said they'd be quite content with that if this relentless progression they were suffering from could just be paused or slowed or leveled off. I mentioned just a moment ago that about 3 months after infusion, there was an abrupt change, like a switch, almost overnight. And the thing that we noticed first was an abrupt increase in energy. Fatigue is the most common symptom that MS patients experience. Not every MS patient does, but it's the most common if you were to talk to hundreds or thousands of MS patients. Fatigue is usually on their list. And it's a profound fatigue. For those listeners that haven't experienced MS themselves personally, if they've come down with a really bad cold, like a really bad one, they'll know that maybe that day or two before they develop a sore throat or sniffles or cough, they feel like they're so wrung out of energy it's hard to stand. That's more similar to the fatigue of what MS patients feel. And they learn to live with it a bit, but it's really difficult. Well, every subject that we talked to said that fatigue lifted. and this is new to us. We haven't seen this kind of phenomenon. An MRI doesn't measure fatigue, and as much as we've tried to look for biomarkers that might be associated with this fatigue and lassitude that's characteristic of MS, we haven't found any marker, any blood test that can help us gauge the fatigue a patient is feeling. So what we did in this study was we used patient-reported outcomes. Those are subjective. They're not truly scientific, but they're important because they speak to the patient's personal experience. We employed a quality of life survey that gets at fatigue, that's been validated, it's widely accepted in our community, and we were seeing improvement on this scale of between 8 to 12 points, which, if you don't know the scale, is a profound improvement. What might make more sense is for me to share the story that these patients, I believe all of them, were having to take naps or two every single day of their lives. They'd get up, they'd be able to walk around, do a couple things for a couple hours, they'd have to take a nap, and then maybe a second nap every day just to maintain energy. Suddenly they didn't need naps at all. And they told us it had been 15, 20 years since they didn't have to take a nap. Several of them told us that they felt their energy levels were the same as before they even got MS. Suddenly they were able to go about their day, be with their family, be effective at work, and maintain their energy and alertness all day long. We had never seen anything like that before. And these are small numbers. So again, it's really important that we keep even, that we employ equipoise, we not make too much of this. But it's an astonishing result to see this level of improvement of fatigue. We did see that, and we continue to see that to this day. And the first subject has been now, well into 1 year past her initial infusion.
That's great. That's all my questions. Thank you so much for speaking with us, Professor Dunn. Thanks so much for listening to today's podcast. This discussion was part of our coverage of the 41st Congress of the European Committee for Treatment and Research in Multiple Sclerosis. To see more coverage of ECTRIMS, head over to VJNeurology.com. And be sure to follow us on X at VJNeurology and subscribe to our podcasts on Spotify, Apple, or Podbean. Until next time.